This is work from our lab, and we've been looking at, we've developed an in vitro CRS model. And the in vitro CRS model is, it has its limitations. And the problem with developing in vivo CRS models is there are even greater limitations for those, in that we use, generally speaking, immunodeficient mice, and immunodeficient mice have no human hematopoiesis, and they have dysfunctional macrophages and monocytes and no T cells. And so the inducing CRS in immunodeficient mice, which is how we really test CAR Ts, has been very problematic. And so there are several papers using very contrived models in immunodeficient mice. It's not clear if this in any way reproduces what's going on in a human being that develops CRS or neurotoxicity. So likewise, and also when you use mice, the experiments are long, costly, and variable. And so we've developed in vitro models to look at, in particular, IL-6 expression. And these in vitro models will have allowed us to test CAR Ts targeting a specific, in one example, CD19 positive targets. And we include human monocytes in these cultures, and then we can measure the production of IL-6. And so we're looking for ways to either genetically modify the T cells or to treat the suspension of CAR Ts, targets, and macrophages in such a way with various drugs that will block CRS or block production of IL-6, but not alter the ability of the CAR Ts to proliferate in response to the target or to kill the target. So this, we've done both genetic screens and small molecule screens, and these screens have resulted in the identification of a number of small molecules that have various interesting effects. For instance, one of the first molecules we discovered was dasatinib, which has been now published by two independent groups showing that dasatinib is a very potent inhibitor and a reversible inhibitor of CAR T cell function. But obviously, we're looking for things that don't block both killing and proliferation of target cells and CRS. We're looking for things that would block CRS but allow the CAR T cells to do their dirty work. And so that's the kind of screens that we've set up, and these screens have been using antibodies to block various signaling pathways, genetically modifying high priority pathways in the T cells using CRISPR, and then to use various intelligently designed drugs to target various pathways that came up in these screens. And one of these pathways has been the PI3 kinase pathway that we identified, and Duvelisib is obviously an inhibitor of PI3 kinase gamma delta pathways. And so that was how this started. And so we also observed, like many other groups observed, that not only Duvelisib, but Jakafi is another candidate that popped out, inhibits the inflammatory response in our in vitro model, and also Duvelisib did this. And so both of these kinds of drugs, JAK inhibitors and PI3 kinase inhibitors, we think are potential drugs that can be used to block the inflammatory immune response that patients develop at the end stages of COVID infection. And so if you can take very severely ill COVID patients and prevent them from dying from this really terrible inflammatory response, then I think you're doing a great service, and this is an unmet medical need. The concern for both drugs is that JAK kinases are important for viral replication, and so the inhibition of, not for viral replication, but for T cell inhibition of infected cells. And so there is some theoretical concerns that inhibiting JAK JAK kinases might enhance viral replication. And so that's being tested in at least 5 large randomized trials in the world for JAK inhibitors. And we are now testing Duvelisib in this same kind of model in severely ill COVID patients, where we're hoping that Duvelisib will not impact viral replication, but will reduce the inflammation associated with COVID and prevent these patients from being intubated or prevent the intubated patients from dying. And that's the theory behind it.